GeoMx DSP. The GeoMx Digital Spatial Profiler or DSP provides morphological context in spatial transcriptomics and spatial proteomics experiments from just one slide. The current trade-off problem. Researchers must choose between spatial information or hyperplex. Now the solution. Nanostring DSP combines in situ visualization with molecular profiling technologies to get the best of both worlds. As you can see, we are getting features of both. Now how does it work? FFPE or fresh frozen tissue sections are stained with imaging reagents and assay probes to detect both RNA and protein targets. Imaging reagents here also called morphology markers. Tissues are imaged with a fluorescent microscope and ROIs are selected. Then selected ROIs are exposed to UV light, thus cleaving the oligonucleotide barcode and releasing it for downstream counting via NGS on an Illumina system. This was all. Thanks for watching.